Okay, good afternoon, everyone. I am Katrina Chapman. Today is December 22nd, 2020. Full disclosure and disclaimer, there's a loud dog outside, so it's not my dog. If it starts yapping again, I can't control it. Today I'm going to be talking about an article that I read for my Genetics Bio 1040 class. The title of the article is Gene Therapy Comes of Age. It discusses a lot of different gene editing techniques. Pretty much 45 years ago, Theodore Friedman proposed that gene-based therapies could be used to help with conditions such as sustaining production for the clotting factor in patients who have hemophilia. It was theorized that this treatment or gene editing could be done through the use of hematopoietic stem cells. So it was just a base theory. It wasn't actually anything put into place back then. Now we fast forward to the 1990s when more research has been done, and finally there was actual gene-based therapy being conducted. These were just initial trials, but they did end up having setbacks. So these clinical trials yielded results that showed serious toxicities with the patients. Basically, these patients were having immune responses to a lot of these gene editing techniques or the viruses that they were using. One of these was the anti-AAV immune response. A lot of patients may have already had been exposed to this AAV virus, and so they were pretty much rejecting the edited gene that was encapsulated within that virus. Now, the article highlights 3 essential tools that are needed for gene editing or for gene therapy. They discuss the adeno-associated viral vectors, the lentiviral vectors, and the gene editing complexes. After the failed initial trials or the setbacks from the initial trials, the scientists decided to go back to the basics, and they wanted to focus on better understanding the target cells and tissues, the viral vectors that they wanted to use to deliver these gene edits, and then they wanted to focus on the diseases that they were trying to treat or create therapies for. So after in-depth research and study, they were able to advance some of these gene editing techniques into different techniques that we may have, or you guys may have heard of in some of your science classes. So I'm going to highlight just a few that were highlighted in the article. There's the ZFNs, which are the zinc finger nucleases. There is TALENs, the transcription activator like effectors nucleases. And then there's CRISPR, which a lot of people have heard of now, the clustered regularly interspaced short palindromic repeats. All of these gene editing techniques or advancements helped simplify the gene editing process. These technologies helped advance the already existing processes that were in place for those initial trials. For example, there is the CAR-T or the chimeric antigen receptor T cell method, and some of these scientists decided to combine the TALENs gene editing method with the CAR-T cell method, which helped to prevent graft versus host disease in patients. All these approaches in gene editing allow for addition, ablation, or correction in a large variety of genetically acquired diseases or genetically inherited diseases. Without a lot of these techniques, we wouldn't be where we are at right now with the advancements in biotechnology. And a lot of these gene editing techniques are not just applied towards humans. They've been successfully applied towards agricultural needs and things like that. It is interesting that this was the article chosen for our review because I was recently watching The Good Doctor, and in one of the episodes, they actually had a patient who was suffering from lack of an immune system, and basically she had to have surgery. I don't remember what kind of surgery she had to have, but they realized that they could conduct a clinical trial on her, and pretty much similar to these, try to do some type of genome editing with her, where she would actually have an immune system and not have to live in her bubble. The patient survived. I don't remember the outcome, but I just thought it was really interesting that it was a gene editing article. But anyways, that's all I have for you guys today. These are the cards that I used. Cue cards. Nothing on the back. Okay, well, thank you for listening and happy holidays.